Hello everyone, how you all? कैसे हैं आप सभी लोग? I hope you all must be having a great time. This is your faculty Ankit and now we will be continuing in our paper number 2, that is unit number 1, which we have already started, that is cell biology and genetics. In the last lecture we have seen cell structure and function. Now this we are going ahead with lecture number 2, and over here we will be focusing upon the synthesis, structure and function of genetic material. Very important, another subtopic of this particular unit, guys. But before moving ahead, let us do a quick recap of what we have seen in the last class. Just wait, Hana. Our last class was about lecture number 1. This is a quick recap which we are seeing. That was about cell structure and function. Just with me go through your notes what you have made, right? And some of you have shown me your notes also. That is a mandatory, because first time in unit number 5 for paper number 1 you have shown me. Now paper number 2 also I have to see, so that I can keep a check how you are writing that. Then I will be giving you a green signal to go ahead. Right. So over here you will be finding that in the last class we understood about cell theories. Quickly go through the classical theory and its scientist names. That particular things you know that what to write, but scientist names you might forget, and modern theory over here. Then after that we understood the cell types, animal and plant, or eukaryotic or prokaryotic. Then we have understood the cell organelles. This is very important, and we started with cell wall, then cell membrane, then mitochondria, then this plastids under this chloroplast, then your ribosome, then your endoplasmic reticulum, then your Golgi apparatus. Structure and function. Take 5 minutes, pause the video and try to see what you have written down in your note. Just go through it. No need to go in detail, but just go through it, right? Now, from the last class, we have understood that over here, first of all, let me show you that over here what topic we are going to pick, and the topic name is synthesis, structure, function of genetic material. Let me show you. This is the syllabus written like this: synthesis, structure, function of genetic material. This we are going to see. Cell cycle is still left, right? And I'll be telling you at the right moment, right time. Don't worry about it. This is my job when I have to teach you this. Right now we have to understand this. That's why I'm saying that you don't have to go unit wise, you don't have to go syllabus line wise. Otherwise it will get confused. You will be getting confused every time you will be reading. Okay, so right now we will be picking up this particular topic over here. And then after understanding certain chromosome structure over here also, then you will be building this cell cycle over here, guys. Okay, the right time I'll be telling you. So this we have covered in the last class, this we have to cover, and this topic we are picking for today, very important: synthesis, structure, function of genetic material. Genetic material, you must all be knowing that this is DNA, RNA, and all these confusion related to DNA, RNA will be cleared today, guys. Today we will be handling all the details of it in this particular lecture with animation I am going to show you. See the question time and again how they are asking you. They're asking you describe the mechanism of nitrate assimilation and synthesis of amino acids in plants. That means how it is happening. The new type of question you have seen in 2025, guys. 2025 examination, you will be finding that how they have clubbed two things. How do plant absorb nitrogen from environment? It is of paper number 1, unit number 5, nitrogen fixation. They are nothing, just asking nitrogen fixation. And nitrogen fixation is written in your unit number 5 of paper 1. And the second, the mechanism of nitrate assimilation and synthesis of amino acid is basically nothing but from you will be understanding your translation and transcription procedure they are asking you. Because from transcription and translation only you get these amino acids, proteins. Protein formation is nothing but amino acid. So that is how they have clubbed these together, and this is one good question in 2025. Apart from that, everything was same. But this is one innovation with respect to the syllabus line they have did. Next question, you will find the structure of DNA. Next, RNA, DNA difference, RNA, DNA components. What are genes? What is genetic material? DNA, RNA, RNA, DNA over here. So everything you will be finding like this, genetic code over here. So everything you are finding with respect to DNA, RNA, they are talking about, and this is what genetic material we have. All right. So till now you are understanding. If I ask you that over here, you have this broad plant cell wall. And if this is a cell wall, that means it is having primary cell wall, secondary cell wall, and middle lamella. This is over here. If I draw like this is over here, plasma membrane attached to it, like this. Like this, you will be finding plasma membrane. Plasma membrane, you have understood different theories. You have understood about the Golgi, endoplasmic reticulum like this. You have understood about the Golgi apparatus like this. You have understood about the ribosome 70S, 80S. Eukaryotic is having 70S or 80S? 80S. You have understood about this particular saucer shaped, bean shaped you will be finding. What is this? Christie, this mitochondria. You have found the chloroplast. Chloroplast like this you will be finding over here. Double membrane again. Thylakoid you will be finding in between. Oh, really sorry guys. It went missing. है ना? अब मैं थोड़ा सा short में बनाऊंगा. I cannot draw everything right now. So this it all got missed. है ना? अभी right now I was making you in front of you only. So that's why it got missed out. Okay, कोई बात नहीं. So I have told you that there was endoplasmic reticulum over here like this. Endoplasmic reticulum. Golgi apparatus. Okay, you have learned about the mitochondria. You have learned about the plastids over here. Thylakoid. Now, the most important part of the cell which we have not discussed till now is this only. This, what is this? What is this? This part. This is nucleus, nothing else. The most important part which you will be finding in the cell. Dot type of structure which in microscope you were seeing, nucleus. And why nucleus is important? Because over here only you will be finding genetic material. Major genetic material. Genetic material, do we only find this genetic material in nucleus, or do we get it in mitochondria and plastids also? In mitochondria also, in plastids, in chloroplast also you get the genetic material I have told you over here. But this is of insignificant amount as we compare it to the nucleus. Nucleus is the major genetic material containing unit you will be finding, organelle you will be finding. So this organelle which we have left in the last class, now we will be understanding it. Its structure we will be understanding, structure and function, and I'll be showing the animation also in today's lecture with respect to this. Okay, so we'll be finding it. Okay guys, now let us see one by one what is the basically structure of this what do we say, this nuclear membrane or this nucleus, right? Let me tell you about this particular part over here. So over here you are having this cell wall, guys. Okay, like this you must understand that this is a cell wall over here. Right, double right now I'm not showing. चलो दिखा देते हैं, double भी दिखा देते हैं over here. Double cell. And this is I am drawing you for for you because you have to draw it in the examination also. This is not for my sake I am drawing. Okay. Now you will be finding that inside that you are having this whole large nucleus, and how this nucleus look like, guys, you try to understand. I'll be drawing a broad type of nucleus over here. थोड़ा सा बड़ा बनाऊंगा, ठीक है? So try to understand how it looks like. You understand, first of all, tell me, this nucleus is a single membrane organelle or a double membrane organelle, and how many organelles you have seen that they are basically double membrane. So like this you will be finding this is your nucleus, and this what I have drawn is a double membrane nucleus, because nucleus are having double membranes. The outer membrane, this is your nucleus membrane. It is outer and inner, and in between them you will be seeing the gaps like this. and these gaps are like nothing but this helps in transporting the material in and out, exchange of material, and it looks like this. ठीक है, प्रोटीन चैनल्स बोलते हैं हम इसे, ऐसे ऐसे दिखते हैं. I'll be showing you this too. Don't worry about it. This like this you have to draw it in the examination also. These are protein ports you will be finding through which the materials will go out and will be basically transporting the material inside and outside it. ठीक है, on the outer surface of nucleus membrane you will be finding that over here ribosomes will be attached. Ribosomes will be attached, and the role of my ribosomes over here, I have told you, either they will be attached in the nucleus membrane or they will be attached over here with respect to the endoplasmic reticulum you are going to find. Like this you will be finding that some ribosomes will be attached to this endoplasmic reticulum, and some ribosomes will be freely available like this in the cytoplasm. This is cytoplasm. What is protoplasm? I hope you can answer what is protoplasm, guys. If not, see the previous lecture carefully. Then you are going to find this is a nucleus. Nucleus membrane is double bound, double membrane. Okay, more information I'll be giving you right now. Just try to understand the structure for now. Now inside, when I'll go inside, what I've found that over here there is one type of condensed material like this. And this condensed material, I have told you, inside the nucleus, it is nucleolus. Nucleolus, very important. And the major role you will be finding of nucleolus is to build rRNA. And rRNA is very important over here. You go rRNA and it will be combining certain proteins and giving you ribosome formation. So the major role of nucleus is to form ribosomes, not rhizobium. I'm really sorry, it is a bacteria. Ribosomes, because these ribosomes you are finding over here, red color things you are finding, it is created inside this nucleus. Inside this nucleus in nucleolus. All right. This particular now fluid inside this you will be finding this fluid over here, guys. This area, this whole area is covered with the fluid, and this fluid type of things we will be calling it as a nucleoplasm. And these nucleoplasms will be containing your enzymes which are needed. Your nucleoplasm will be containing this nucleolus. Your nucleoplasm will be containing this chromatin fiber, which I am going to show you, which contains, where is DNA right now? I have not shown you any DNA. DNA will come now, guys. DNA will come now. And how? See this particular DNA is like this. Like this. In this form, DNA is present. In this form. Okay. So what do we call this particular part is chromatin fiber. Chromatin fiber. Not chromatin, not chromosome right now, but chromatin fiber. Okay, and it is spread like this inside this nucleus. Where is DNA? DNA is present in this chromatin fiber only. In this fiber only you will be it will be basically having DNA, nothing else. Right, and that we have to explore. Right now you just see that the structure of nucleus contain a nucleus membrane, nucleolus, nucleoplasm, and this chromatin fiber over here. And this chromatin fiber is containing your DNA, the important genetic material which is containing our codes. Even if I don't know you, I can create you by the fragment of this DNA. Right? And then you will be finding their job is containing the code, and that code will be taken up by mRNA. It will be translating it into protein with the help of these ribosomes. With the help of these ribosomes, and it will be transported through now endoplasmic reticulum, going to this Golgi bodies, and then doing their dedicated function, targeting wherever either they want to move out or they want to build some internal mechanism over here. I hope this you are understanding, guys. Now let us explore more about this in the form of, let me show you the diagram now. Now you will be understanding the diagram. Okay. The most important discovery which has been done with respect to nucleus was Robert Brown. Some discovery A.V. Leeuwenhoek has also done. But Robert Brown you will be remembering over here, guys. Okay. Try to see. This is your nuclear pores. Pores I have shown you in what picture? I have shown you the picture which I have drawn, right? वो अभी मैंने आपके सामने बनाया था, वो चला गया जैसे ही मैंने आगे किया वो, ठीक है. It was a temporary screen I was making on, ठीक है, but you have to make it, है ना. On this outer surface you are seeing these ribosomes are attached. Double membrane over here, the outer membrane ribosomes are attached over here. Then these are endoplasmic reticulum, and over here also ribosomes are attached. Inside this you will be finding nucleoplasm and chromatin fiber, and this is a nucleolus inside that also. And this is a nuclear envelope, nothing but that thing only you will be finding. So first observed by Robert Brown 1837 in a flowering plants, you will be finding Robert Brown finded this nucleus. Number of nuclei per cell, single nucleus per cell you will be finding. In one cell you will be finding one nucleus. This is the storehouse of our information, you can say, right? Plant cells more than one nucleus can also be found. We can call it sinusoids. Like over here in certain algae, Rhizopus, Vaucheria, you can remember these exceptions. If in animal cells multiple nuclei are being found, it is called syncytia. Striated muscle cells of higher animals you can see. But these are exceptions, upward exceptions you have to remember. Just okay, these things also happen. Position in the cell you will be finding sometimes central position they occupy, sometimes surrounded by cytoplasm, separated by a nuclear envelope you will be finding over here. Morphology of nucleus you will be finding that their shape variation happened from spherical, ellipsoid, flattened, right? These are just nothing but a simple information which you will be finding over here. The major thing which is coming right now is the components of nucleus. Now you don't have to do anything new, you don't have to remember anything new. What I have discussed, nuclear membrane, you have to extrapolate it. Extrapolate means double membrane, and it is a semi-permeable because it is having nuclear pores. The pores I have shown you like this, protein pores you will be finding. It is perinuclear space. So like this I have drawn. So there is a double membrane. So in between part, in mitochondria we were calling it perimitochondrial, perichloroplastidial, and over here if this is a nuclear double membrane, then perinuclear space. I have told you that there are three organelles only which are having double membrane. Plastid, that is chloroplast. Second is mitochondria. Third is your nucleus, right? Appearance, thin line under light microscope between nucleus and cytoplasm. Provides a pathway for material support between nucleus and cytoplasm. Over here you will be finding, okay? Because it provides a function of nuclear envelope and nuclear membrane को आप देखोगे. One is a nucleoplasm. Nucleoplasm is the liquid present inside the nucleus. The cytoplasm is the liquid present inside the cell, right? Protoplasm will be including both of them. So nucleoplasm is a fluid filling the nuclear space around chromosomes and nucleolus. Escapes if nucleus is punctured, it will be escaping, and this is also known as karyolymph. Composition, it is rich in acidic proteins, contains RNA rich in adenine and uracil. That inside this RNA will be present, guys. rRNA, tRNA, will be seeing it all. Okay, contain various enzymes also in order to function the functioning of this nucleus. Okay, their function is to act as a medium for metabolic activities of the nucleus. Site for enzymatic reactions also you will be finding over here. Then you are finding this nucleolus. Nucleolus first described by Fontana. Important to remember. Okay, Fontana discovered nucleolus. It is a spherical, relatively large body in the nucleus. Numbers varies per species, and number of chromosome sets also you will be finding. Usually one nucleolus per haploid chromosome set we get. In this particular thing you will be getting one special region known as nucleolar organizing regions, NORs. That I'll be showing you later on, but you remember that NOR is also present. When I'll be teaching you about chromosomes like this, I'll be telling you where is this NOR region and from where this nucleolus emerged. Okay, over there you will be remembering NOR. Right now not needed. Behavior during cell cycle. Cell cycle we have to study. Again we will be seeing this. Disappears during cell division. When it disappears I'll be telling you. Reappears in daughter cells after division. This also I'll tell you. Exception, persistent nucleolus in Spirogyra and Euglena you will be finding. So cell cycle you will be finding that this nucleus will be disappearing during the division and will be reappearing when the division has taken place. When it happened, how it happened, I'll be showing you by animation, guys. Don't worry about it. Okay. Function is synthesis of rRNA, what I've told you, and formation of a ribosome. Over here you will be finding two important function of nucleus, because ultimately our body is working through protein synthesis only, and proteins are being synthesized by ribosomes, and ribosomes are created by this nucleus. So that's why nucleus is very important. See this chromatin over here now. Chromatin, I have told you about the chromatin fiber. This chromatin fiber is a dark stained material in nucleus. Combination of DNA, histone proteins and other proteins also you will be finding. Structure during interphase. Interphase, when cell cycle, I'll be telling you there is one phase known as interphase. There is a loosely coiled strands called chromatin reticulum. There I'll be telling you. Okay, that's why I'm saying you this whole unit is connected. Okay, several answers you will be getting later on. Structure during cell division. Again, cell division, you will be finding how this converted into chromosome. Why I have told you that chromatin is different and chromosomes are different. Not particularly different, but structurally they will be different. Chromatin you will be finding like this, guys, like this, very loosely over here. But chromosome is a stage where chromosomes particularly you must have seen like this you must have seen, right? Like this, in pairs you must have seen like this, so this stage basically comes during the metaphase you will be finding, or during the cell cycle when I will be telling you mitosis, meiosis. Then it appears like this condensed material over here. So this will be converted like this during the cell division process, and when cell division will be happening, then we call them as chromosomes, because they will appear like this in a particular plate, metaphase plate you are going to find like this. And this I'll be going to tell you later on, and I'll also show you too. Again I'm telling you, okay? Functions. Their function is to package DNA into smaller volume to fit inside the nucleus, strengthens DNA for mitosis and meiosis, controls gene expression and DNA replication also. Very important. Okay? So these, when they will be asking you about the structure of nucleus now, you will be drawing the diagram I have drawn you by my hand. That means it can be made up, और ऐसे diagram नहीं बना पाओगे आप ये वाले, ठीक है? वैसे वाला diagram बनाना. And then you have to explain these four structures over here: chromatin, nucleus, nucleoplasm and nuclear membrane. ठीक है? This you can explain. Simple 10 marker or 15 marker it will come. You have to fill 2 pages or 3 pages. One diagram and these explanation you will be giving. Now again you can see the same diagram over here. Pores are present, nuclear membrane is there, and again always you have understood that membrane is containing this phospholipids. This is nothing but a representation of phospholipids. ठीक है, देखो phospholipid भी लिखा है. ये perinuclear space है. इसके बाहर का outer membrane, ये वाला outer membrane में you will be finding ribosomes also. Then you are having this nucleolus, nucleoplasm and chromatin over here you are getting like this. ठीक है, ये आपका chromatin present है. Understanding? Now let us have the understanding of this DNA, guys. अब आप understanding लो DNA की, RNA की, structure समझेंगे हम DNA का, RNA का. We'll be understanding the whole structure. Don't worry about it. And functions are related to that. ठीक है. First of all, I have told you this. From where we have understood, you try to understand. This is the cell, plant cell. This is the nucleus. This is the chromatin fiber present over here. Now I am taking this chromatin fiber over here. Chromatin fiber looks like something like this, fiber like of structure. Then I am picking one fragment over here. Then what you will be finding in the cell division phase, you will be finding that these will be arranged like this. Humans have 23 pairs. This is one pair, like this you will be finding. ऐसे connected होते हैं, ठीक है? Like this you will be finding, ठीक है? Like this. One particular chromosome is this, second particular chromosome is this, and this is calling one pair, two pair. In peas you will be finding 14, 20. Wheat you will be finding, and maize में wheat आपको मिलेगा 20. ठीक है? Likewise you are going to find these chromosomes, and these chromosomes are representing nothing but a species, a particular species. 23 pairs in Homo sapiens you will be finding. Now what I am doing over here is I am picking this particular chromosome, this which we have seen over here. I am seeing this and opening up. Over here this DNA is coiled up, as you must have read. Like this DNA is coiled up in condensed manner like this. But we will be opening it up. So it will look like this. We will more open it up and it will look like this, guys. Like this coiling has happened of this DNA. What is DNA then? This DNA is this. This is a chromosome. When we will be opening up this chromosome, we will be finding that whole DNA is being coiled up with the help of this histone proteins. These are proteins on which these particular things are coiled up. And what is histone? Later on we'll be understanding when we'll be understanding about the chromosomes. Right now our objective is DNA. So these are histone proteins on which this DNA is coiled up. This white thing you are finding, this is nothing but DNA. What is a gene then? There is a difference. I am telling you clearly. A lot of people get stuck in this. Well, for a lot of people, you will be finding they will not be able to tell you the difference between chromosome, DNA, and gene. Gene is the basically functional part over here now. Okay, I am telling you from the non-bio student also, those who have not done biology, but this you have to do in your GS3 anyway also. So it is nothing that we are learning new over here, right? This you already obviously have to do GS3, but if you are having this agriculture optional, you have taken my course, then you don't have to worry about GS3. So chromosome is this large unit, condensed unit over here. It will be opening up, and this is the DNA coiling only, and DNA coiling has happened with respect to histones. Gene are the functional segment over here. Suppose this function is related to this particular part is related to my eye color. So this becomes a gene which is containing a code for a particular expression in a body, either somatic expression or non-somatic sexual expression, you can say. That I'll telling you later on what is somatic, what is sexual over here, right? Right now just understand chromosome, you have seen DNA, you have seen this all is DNA coiled up in histones. Histone you will be understanding later on, and gene is the functional part of this DNA. So gene is not everything. Every DNA. Gene is the functional part in DNA because DNA is containing non-coding segments also. This is non-coding. But this over here is coding, and this is coding for my hair color. This is coding for my eye color. This is coding for my sexual characters. Understanding? So this is a DNA you will be finding like this. Let me show you also. This is the human chromosomes you will be finding like this. And 23 pairs in maize, different pairs in peas, 14 chromosomes you will be finding. So like this genes are attached. This is one pair of chromosome you are seeing. Now this coiling I am opening it up. देखो यहाँ से देखो नीचे से ये coil open up करी और इस हिसाब से this DNA is coiled up. जब आप इसे और open up करोगे तो these ball like shape you are finding, these are nothing but proteins, histone proteins. You have to remember histone, nothing else. क्या है histone, कैसे बनता है histone, वो बाद में देखेंगे. Then you will be finding that when you will be opening up more, then this coiling has happened in this double helical shape like this. Double helical shape you will be finding, guys. Like this you are going to find, and this is nothing but DNA. So let us see one by one what you have to see in DNA. Understand the most important thing now, guys, related to the evolution of DNA structure, that how we found this DNA structure over here. Alright, so different experiment, different scientists were involved over here. Till now question has not come explaining the scientist's name and their contribution in finding the DNA structure. But in other areas, in other topics, the scientists become important. I'll be telling you in different units, right? But from here, from this particular topic, question has not come in this way, but it will come soon. I am telling you. Okay, so the most important things you will be understanding that in order to identify this DNA, how scientists evolve this theory, or how scientists come up with this particular DNA structure. So this is a very important and interesting to know that over here, how we got this DNA, the most important molecule, how we have got to know about it. So there are number of experiments which are involved over here, guys. I'll be telling you about these experiments in one go. Otherwise, people have made it a bit complicated. Other people, I am telling you, in agriculture, nobody teaches this also, right? In other examination also, if you will be finding that it is a bit complicated. Agriculture में तो कोई पढ़ाता ही नहीं है इन सब चीजों को, ठीक है? तो you will be finding Griffith experiment over here. One Griffith experiment. Scientist was Griffith, right? He did one experiment in 1928 and got to know about something, that there is something which is getting transferred. What is this? He didn't know right now. But later on, one more experiment was continued by, you will be finding three scientists' names over here: Avery, MacLeod, and McCarthy experiment. And this happened in 1933 to 34 around, you will be finding that they will be doing this experiment. Sorry, change this, 1944 you will be finding over here, right? Really sorry. 1944, this experiment was done by Avery McCarty. Basically continued the experiment which Griffith has done, and the questions which were unanswered in the Griffith experiment were answered by this particular part. One more thing was done again in the contribution to this DNA structure. Very important contribution was given by Hershey and Martha Chase experiment. Hershey and Martha Chase experiment, and this happened in 1952. And then finally, the golden module, because of which we know the double helical structure of the DNA, everything which we know about DNA over here, you will be finding it was culminated by Watson and Crick model of DNA. Though these were not independent experiments, you try to understand. Experiments started from Griffith over here. Now they were evolving more things that what it could be. So all these experiments, the contribution of Griffith, contribution of Avery, Hershey and Chase, ultimately Watson and Crick led to our understanding of DNA structure, the most important molecule, because of which in present day we can utilize CRISPR and all these biotech technologies, which you will be understanding also in unit number 3 CTech, right? So Watson and Crick model, this has been asked already. And whatever we are going to study about DNA is basically Watson and Crick only. But over here also you have to know about these experiments, right? Over here, what are the shortcomings? What were their experiments basically? So that you can write it in 150 words or 200 words in the examination. I'll be telling you, and you will be remembering that too. So first of all, see, these experiments are important with respect to DNA. Findings: Griffith, Avery, McCarty, हर्शी एंड चेज एक्सपेरिमेंट 1952. और ये याद रखना पड़ेगा, एक दो बार रिवाइज करोगे मेरे साथी, और एनरिचमेंट में व्हेन यू विल बी एंगेजिंग विद मी, देन आई विल बी मेकिंग यू रिवाइज इफ यू आर डूइंग इट लाइव. अदरवाइज फायदा नहीं होगा आपको, राइट? मतलब फायदा होगा लेकिन लेटर ऑन भी, बट ट्राई टू डू इट in the live mode also, right? Now see the Griffith experiment first of all, guys. Griffith experiment, very important experiment. Griffith experiment, and this Griffith experiment is also known as transforming over here principle, or based upon this transforming principle, right? What is this transforming principle? I'll be telling you. The major thing which Griffith has taken was basically, he took one culture, he took the culture means where these bacteria will be grown, lab grown by him only. So he grew some over here bacteria, important bacteria which causes a pneumonia, guys, and that is Streptococcus pneumonia. So spelling I'll be telling you through the PPT also, but just take it over here. Streptococcus pneumonia. Okay, exact spelling I'll be showing you in the PPT. Don't worry about it. So when he grew these cultures, what he got to know that there are two types of bacteria which are being grown over here. Like one type of colony of bacteria which is formed is smooth type, and one type of colony which was grown over here automatically was basically R type or rough type. Or this, he termed it as a rough strain of this particular bacteria and smooth strain of this particular bacteria. Now the important thing about over here, one is R strain and another one is S strain. Now there is some characteristics with respect to this, that S strain, the smooth shiny bacteria, are basically virulent, means they cause disease when they will be transferred to any organisms. When these bacteria will be transferred to any organisms, they will be causing disease. And what disease? Pneumonia. Whereas a rough strain is avirulent. Avirulent over here. And what it will be causing? It will be a non-disease causing bacteria. Means if you will be putting it inside any organism, it will not cause any pneumonia or it will not cause any disease, right? So avirulent and virulent type of strain he has got over here. Now the experiment starts, guys, over here. What he will be doing over here, he will be taking a mice. Okay, he'll be taking a mice over here. So there will be 4 cases which he will be engaging with. 4 cases means, first case will be where he will be taking this R strain. What will happen if I take R strain and give it to mice? Will he be living or he will get this disease and die? you will be finding R strain is avirulent. That means it will not be causing disease in mice. Automatically he will be living over here. So you will not be finding that he will be living over here. He will not be killed. So हम इसको tick mark कर लेते हैं, right? That he will live. Second, he took S strain. S strain means virulent one. So when he gave it to mice, what will happen, guys? Automatically he will get the mice will get the disease, and automatically he died in his experiment. So I am putting it cross. That means he died. This is not the cause of concern for him. This was as expected. What he will be doing next is a third case he will be taking, where he will be taking this S strain and will be killing the bacteria itself. The will be basically giving it heat, right? Heat killed S strain over here. This was the name of this particular thing you will be finding. Heat killed means that this S strain he heated first, and the virulent type of thing which was there, it got neutralized. That means it became avirulent, right? So you will be finding this name as heat killed S strain. Heat killed S strain. That means what I have done with this S strain, I have heated it and killed the virulent part of it. Automatically, what will happen when you will be giving it to mice? This is the heat killed S strain over here. So it is avirulent. When it is avirulent, the mice will live. This is also not the cause of concern. Cause of concern will come now. What he did, that he mixed the heat killed S strain and with R strain, he mixed these two over here. Understanding? I hope you can see it, guys. Let me just, I'll be showing you also, don't worry about it. Third case is heat killed S strain. Mice will be killed over, mice will not be killed over here because this is the heat strain, heat killed S strain. But when I am mixing this heat killed over here, heat killed S strain, which becomes avirulent after getting heated, and R strain, which is already avirulent. When I mix this or when he makes this and gave it to mice, what you will be finding, that mice died. How it happened? This is the cause of concern, that how it had happened. So that means he thought that something was getting passed from these two, something has passed, and obviously if the mice has been killed, that obviously the virulent part must have transferred to the R strain. The question is what has transferred. He was not able to understand what has transferred over here. So what he will be doing, he was taking it as that he was thinking that it must be some protein which has transferred from S strain to R strain in the fourth case. Understanding over here? Let me, अगर एक बार मेरे को रुक जाओ, आप देखो पूरा इसको. Now see, I hope you can see the cases. 1, 2, 3, 4. I have removed my the video for a while. Right? Just see. This R heat killed S strain, mice was alive. Whereas heat killed S strain mixed with R strain, mice is dead. So that means something has been transferred over here. He was thinking that there is some protein which has been transferred. Right? So the question was unanswered over here. And what I'll show you the Griffith experiment now. Let me open up the video. Yes, guys. Now see over here. Griffith experiment. 1928, landmark experiment in molecular biology. You will be finding that the name is Frederick Griffith. Though it didn't reveal the structure of DNA, but it laid the foundation for later discoveries you will be finding, including DNA's role in as the genetic material and ultimately its structure over here. So genetic material, that means something is being transferred. What is that material? This DNA over here. But right now he could not come to the conclusion. It was in conclusion you will be finding. So he took S strain and R strain, this like this only you will be finding. And the spelling which I was showing you, Streptococcus pneumonia. Please guys, correct the spelling over here, and you have to learn this particular bacteria also. This is the disease causing bacteria which he grew as S strain and R strain. R strain is a non-disease causing, and S strain is virulent and non-virulent you will be finding. 4 cases, 1, 2, 3, 4 cases. The question was 3 cases were as expected, but as with the fourth case, heat killed S strain and live R strain. What happened? Mouse dies, and this was the cause of concern over here. So when heat killed S strain were mixed with live R strain bacteria and injected into mice, the mice died. That means live S strain bacteria were recovered from the dead mice, right? And this means that something has transformed, and that's why he gave this transforming principle which I was telling you about over here. Transforming principle. Why? That something must have transformed from this heat killed bacteria to this particular non-virulent R strain into a virulent S strain. Ab S strain and R strain will not confuse you. Write it very simple, guys. So simply you write it, right? So in this way, you will be writing that this was inconclusive, but it was important in DNA discovery. Though Griffith did not know the chemical nature of the transforming principle, but he suggested that something has been carried out, something has been carried, heritable information that could transform the bacteria over here. Now, in further 1944, you will be finding Avery, MacLeod, McCarty extended Griffith's work and showed the transforming principle was a DNA. So who will be now highlighting his work that it was DNA? So you will be finding over here Avery, MacLeod and McCarty experiment of 1944. Now they will be extending this experiment, guys. What they will be doing over here? What you will be finding over here, that cause of concern was this heat killed bacteria. Heat killed S strain. So what they will be doing, heat killed Streptococcus pneumonia over here, which has been made avirulent by killing the bacteria, है ना? So what they will be doing over here is they will be taking this bacteria over here and will be putting or will be making 3 cases. And in these 3 cases, what they will be doing, in one of the case where they will be taking this particular bacteria, heat killed S strain bacteria, bar nahi bolunga, but this bacteria they have taken over here, and will be giving one enzyme known as protease. In the second case, they will be giving one enzyme known as RNase, and in the third case, they will be giving one enzyme DNase. Understanding? That means in the first case, he mixed the test tube with the heat killed S strain and protease enzyme, then RNase enzyme, individually or independently DNA enzyme. Okay, so what these things will be doing, protease basically kill the protein part of the bacteria, protein part. RNase destroys the RNA part and DNA destroy the DNA part over here. So in first experiment, when he mixed the particular strain with protease, that means protein part has been killed. But still he injected this to mice now, where protease has been done, protease has been given. Okay, now heat killed S strain bacteria was given to mice in the first case. What will happen? What do you think what will happen? Will the mice be alive, or it will be getting this disease and will be killed? Right, and over here it is mixed with over here R strain 2, है ना? R strain was live bacteria. ठीक है, ये तो चल ही रहा है. तो ये दोनों इन्होंने छेड़छाड़ किसके साथ करी? Where they have done the changes over here. Heat killed S strain by putting protease. That means they have destroyed the protein part of the heat killed S strain. So what happened over here when they mixed these two and given to mice? What you will be finding that the mice was killed over here. That means they understood that it is not the protein which is affecting, or it is not the protein which is passing on, क्योंकि protein को तो इन्होंने destroy कर दिया था. अगर protein से, if protein must be transferring this part to the other organism, to the R strain over here, then it must not have shown its result. It must not have been diseased over here, the mice, right? Whereas then they take the second case, and the second case rRNAs they mix, and then they put it into the live R strain 2, and then they gave it to mice. What will happen over here? At this point of time, you will be finding again the mice was killed. That means it was also not RNA where this information was passing off, the disease causing information. That means there was something else. Then when they saw the third case where they destroyed the DNA part, you will be finding when it will be mixed with live R strain, what is going to happen when it will be fed to mice, guys? You will be finding that in this case mice got alive. That means the information which was passed on was because of this DNA, and that is how they came up with that DNA is a genetic material, right? Because of this the information was getting passed. That is how they confirmed over here. Let me show you the content now over here, guys. Avery, MacLeod, McCarty experiment 1944. Full names you can see over here, no need to study. Now what transformed it? They wanted to find the transforming principle responsible for transferring genetic information in Streptococcus pneumoniae. What they did, they test whether transforming principle is a DNA, RNA or protein, and with the help of these 3 cases they took, protease, RNase and DNase cases over here, which destroys DNA, RNA and protein, self-explanatory. What they will be doing? They will be using the Griffith's earlier observation that heat-killed virulent S bacteria could transform non-virulent bacteria into virulent ones. So they will be taking one strand of heat-killed S-strain bacteria and will be giving these particular enzymes and then mixing with the live R-strain bacteria and then observing it in mice. What is going to happen over here? Protease, protein destroyed. This disease passed? Yes, it passed. RNAs, this disease passed, transformation occurred. But when DNA, one case they have took, they will be finding that no disease was passed because DNA was destroyed. That means DNA was that thing that was transforming over there. Right? So only DNA's treatment blocked the transformation. Therefore, the transforming principle is DNA. Understanding very simple, very easy if you are understanding over here. ठीक है, तो Griffith experiment you have understood, Avody, MacLeod and McCarthy experiment you have understood. Now you go ahead and understand the Hershey and Chase experiment. This is also good over here, guys. Hershey and Chase experiment. Very important this is, guys, and a bit difficult to understand. Difficult नहीं, complicated है और कुछ नहीं. यहाँ पर Hershey and Chase, Martha Chase and Hershey, the name, full name, we will be seeing it, not important, but Hershey and Chase, what they did over here, they basically objective was to find that whether protein also passes on its genetic material or not. This is happening at the time period of 1952. Now, right now we know this information that protein is not a genetic information passer, or it is not a genetic material. Genetic material is DNA, that we know. But at that point of time, the confusion was regarding protein also, that whether protein also transfers the genetic material or not. And also they wanted to confirm, as per the Avery, MacLeod, McCarthy experiment, that DNA was that the transforming principle. So they want to confirm DNA also. So they took over here two things. One, a virus, and one, a bacteria over here. The virus was basically taken by them is bacteriophage. So don't get confused. The name is bacteriophage. Okay? But it is a virus. Now bacteriophage, basically you will be finding it looks like this. Like this you will be finding the structure look like this, right? Not important. But over here what is important, that bacteriophage virus is a special kind of virus which basically infects or kills the bacteria. Right, so they chose a virus over here and they chose a bacteria over here. And what bacteria they chose over here? The most experimented bacteria, that is E. coli. What will happen when this bacteriophage will be given to this E. coli? Automatically this bacteriophage is basically, his feature is to kill bacteria. So when bacteriophage will be going to E. coli, it will be disrupting its functioning, killing the E. coli bacteria, right? So this is, they wanted to understand, but they understood it in a very different manner. How they understood? Because keep in mind that they wanted to identify whether protein also transmits something or not. So what they will be doing over here, this is basically, if this is your, now this is a bacteriophage where you will be finding this is a DNA over here, right? This is a DNA of this particular bacteriophage. This is a virus, right? And over here, this is a protein coating over here on it. This is a protein coat you will be finding. This coat is of caspid बोलते हैं हम इसे. ठीक है? It is a protein coat you are going to find. ठीक है? And this is its viral injecting. This is the way they will be injecting over here, guys. Okay, now they will be feeding on this E. coli bacteria. What it will be doing? It will be injecting this DNA over here in E. coli. And what this E. coli will be doing? Making the several copies over here, and further E. coli, further this particular bacteriophages will become alive over here and will disrupt this particular E. coli. Right? This is how the virus works. So what they have done over here, that to identify, first of all, they will be growing this bacteriophage in two mediums, guys. Two mediums, two culture mediums over here, they will be growing it. Okay. Again, over here they will be taking this. In one medium, what they will be doing, that they will be coating or reacting this particular bacteriophages over here, these bacteriophages which you are getting. Virus को इसमें डाल रहे हैं दोनों में. ठीक है. At one coating they will be doing of the protein coating, and protein coating they will be doing with the help of radioactive sulfur. They will be utilizing radioactive sulfur over here. Means what they will be doing, they will be making this protein coat radioactive in nature by adding radioactive sulfur to this protein coat. Okay? So in this medium, this has become radioactive sulfur, this protein coat. है ना? In the second medium, what they will be doing, that they will be making this DNA of this virulent virus, of this virus over here, that is bacteriophage, they will be making the DNA radioactive by a radioactive element, that is phosphorus. So they will be mixing this radioactive phosphorus and making this DNA over here radioactive in nature. What you will be finding, that means now there are two type of bacteria we are having from this A culture and from this B culture. From A culture we have that type of viruses that are having radioactive protein coat, whereas from B culture you will be finding we have certain kind of viruses which have a radioactive phosphorus DNA. The DNA is radioactive over here, different things over here, DNA radioactive, over here protein radioactive. Now if they will reproduce, if you think this A virus is reproducing, what you will be finding, this radioactivity will be passed on to the next generation or not. Next generation or not, it will be passed or not over here. The protein code, the radioactive protein code will be passed to next generation or not. What do you think? Whereas this radioactive DNA which we have made, if we will be reproducing it, what will you find over here? Will they be passing this particular DNA, radioactive DNA, to the further generation or not? Right now you must know that DNA is basically your genetic material heredity. Because of what happens, this DNA, so DNA will be passing this for radioactive phosphorus to the next generation and will be showing the radioactive DNA. Whereas protein will not be passed to the next generation because it is not the genetic material, right? So it will not be having the radioactive protein coat. This you must be understanding, but this has been highlighted by Hershey and Chase by this experiment. What they will be doing now, they will be doing three processes. They will be highlighting this by three processes. One is infection. Second is blending. Third is centrifugation. Centrifugation means what will be happening in the first case, guys? Try to understand over here. What will be happening in the first case? In the first case, they will be basically infecting. Suppose these are the two test tubes. E. coli is present over here. E. coli is present over here. They will be basically injecting or infecting this particular E. coli with the A type of culture over here, whose protein coat was radioactive, and B type whose basically DNA was radioactive, है ना? And when they will be infecting over here with this, basically what they will be doing, then blending will be happening amongst these. Alright, and centrifugation means then they will be rotating this particular, with the help of some instrument, they will be rotating this particular thing. What will be happening by that? You will be finding that the two layer will be separated over here. Let me show you. See this particular over here. Alfred Hershey and Martha Chase over here, right? To confirm whether DNA or protein is the genetic material that viruses transfer into bacteria. What they will be taking, bacteriophage, a virus which I have told you which affect E. coli bacteria. Two things, bacteria and a virus over here they have taken. What they will be doing, they will be making two cases. One case where DNA they will be marking it with the radioactive phosphorus. Protein they will be marking it with the radioactive sulfur. Over here what will be happening, two sets of bacteriophages were prepared. This I have told you. Now each labeled phage was allowed to infect with E. coli bacteria. That means infection will be happening in the first process. Then after infection, the mixture was agitated in a blender. That means blending will be happening at the second stage to separate the phage protein coats from the bacterial cells. What they do, what they mean by this, that this, suppose this is a bacteria, E. coli, and this particular virus has injected. Now the virus, how it inject, basically the protein coat do not transmit itself into the E. coli. Basically this DNA material will be gone over here. So on the surface what you will be finding, that these protein coat of the bacteriophage will be there like this. So by blending what will be happening, that you will be separating this protein coat. This protein coat will be moving out when you will be like blending it over here. So understand, it was agitated in a blender to separate the phage protein coats from the bacterial cells. Right, the protein coat has been moved away now. Now fourth one is the solution was now centrifuged to separate the heavier bacterial cells known as pellet from the lighter phage coats known as supernatant. Nothing over here, don't worry about it. Basically what they will be doing now in the test tube, they will be utilizing the centrifuges. When it's what centrifuges does, centrifuge, देखो centrifuge अगर आपने देखा होगा या नहीं देखा होगा, it will be like this. These are suppose certain test tubes. right? Certain test tubes you will be seeing over here horizontally. So this will move, rotate like this. So what will be happening? Denser particles will be coming over here in the center, whereas the lighter particle will be moving out at the outer part over here, right? Like this. So the center part is known as heavy bacterial cells are known as pellet, whereas the outer particles which are lighter known as supernatant. So don't worry about it. Like this you can show in the test tube also. This is pellet, this is supernatant. ठीक है. Now when you will be observing this, देखो observe करो आप, ठीक है. You have two processes. One test tube you are having over here, and second test tube you are having over here. What you have over here after the centrifugation process, that in this you must have added the E. coli, or you must have added the protein radioactive, whereas the protein was radioactive and this DNA was radioactive. This is the E. coli, because this is heavier, so it will be coming over here, and this is the E. coli in both the cases, and this is the virus over here, and this is the virus over here you will be finding. Okay, so what you will be seeing, guys, that this virus which was having protein radioactive over here, you will be finding that this E. coli was not having any radioactivity present in this. That means this protein does not transmit radioactive. If protein would have transmitted or something to the E. coli, then E. coli culture will be having, or these E. coli must have had certain protein coating in them, also radioactive protein coating in them. Whereas you will be finding the virus over here which was DNA radioactive, whose DNA was radioactive, you will be finding that they have made the colony in E. coli, where you will be finding the bacteriophages which are having DNA radioactive too. That means DNA is the material which is transferring its genetic material. That is what we have known from this experiment. So pDNA found inside bacterial cells in pellet. S sulfur protein labeled, you just understand protein labeled, found in the supernatant outside bacterial cells. That means only DNA entered the bacterial cells and directed the production of new phages. Therefore, DNA is a genetic material, not a protein. Again, you try to understand, there might be some confusion. I'll be clearing out this confusion over here. You have, first of all, you have created two culture mediums for the experiment. You have taken two test tubes also for the experiment, suppose over here. Okay, like this you have taken. जिसको समझ में आ गया, they can like proceed ahead, but this is some of the people should know it clearly, right? Over here, what you will be finding, one particular part, what they tried to do, that they wanted to coat, they wanted to highlight protein. So how they highlighted protein? With the help of radioactive sulfur. Second part, they wanted to highlight the DNA of this virus. तो यहाँ पर हम वायरस के साथ छेड़छाड़ कर रहे हैं. वायरस का नाम क्या है? बैक्टीरियोफेज. So over here they highlighted the DNA by radioactive phosphorus. ठीक है, now the DNA is ready over here, red colored. Now over here in this E. coli is present. E. coli is present. What you will be doing over here, these bacteria you will be sending, these viruses you will be sending these test tubes. So one A virus will be going to this particular, B will be going to this particular. यहाँ तक कोई दिक्कत नहीं होनी चाहिए. Now when this virus will be going over here, protein coat, protein radioactive, like this, they will be attaching the protein coat over here and injecting the DNA material over here. That means in this E. coli there will be several copies of these bacteriophages will emerge. Same will be happening over here. होगा या नहीं होगा? This protein coat will remain outside, and inside this several viral copies will be coming over here. Inside bacteriophages will be coming. Now second is blending. First you did the infection. Second is blending. When you will be doing the blending, this protein will get detached, right? This coatings will detached over here. They will get free. And third, you will be doing the centrifugal force. You will be doing this centrifuges. What will be happening because of that? You will be finding that over here, let me show. Over here, there will be accumulation of E. coli. There will be accumulation of protein coat over here, and E. coli. Now, what they have noticed over here, that these E. coli are not having the protein, radioactive protein over here. That means it has not been transferred from this virus to this particular E. coli. ई कोलाई में जो वायरसेस आ रहे हैं, जो रिप्रोड्यूस हो रहे हैं, कहने का मतलब ये है that viruses which has been reproduced inside E. coli were not showing any radioactive protein code, whereas the E. coli over here, whereas the viruses which were produced inside this B case E. coli, there we have seen the radioactive DNA. That means the radioactive DNA was passed on by this genetic material called DNA. ठीक है, this is what you have to know from here, and this way you can write it in the easy way in the examination. Not more than this will be asked. If this question will be asked, they must be asking about the 3 experiments altogether, not one experiment they will be focusing upon. ठीक है, so significance was this experiments provided clear experimental proof that DNA carries genetic material information, resolving the long-standing debate over the genetic material's identity. It strongly supported the idea that DNA, not protein, is responsible for heredity. Very important. And in summary, you will be finding that DNA, phosphorus inside bacteria, genetic material. Protein, sulfur remain outside bacteria, not genetic material. ठीक है, so I hope you have understood the Griffith experiment regarding the R strain, H strain. Over here the 3 enzyme protease, RNAs, DNAs, and over here the bacteriophage plus E. coli experiment of Hershey and Chase. I hope you have got the better idea, guys, over here. Now some of the students are finding more content with respect to it. You will be wasting your time, guys. Don't find extra content with respect to this particular topic now. This is over. Right, more than this never be asked in the examination, I am telling you. ठीक है, इतना हो गया. अब the most important part which we have to see is a Watson and Crick model, guys. अच्छा, what we have seen in this Hershey and Chase, na, this is also known as transduction principles. Transduction principle. Whereas Griffith experiment, transforming principle. Just name, nothing else. Transforming principle, right? See now, whatever we know about this DNA in detail information, basically the Watson and Crick model is one of the strongest base which they have given for the DNA. And what they have gave over here, that they have given DNA is a double helical structure. Like this DNA is over here. This structure, the double helical, which you all must have seen, has been given by this Watson and Crick. So structure details, I'll be telling you: double helix, backbone, base pairing, anti-parallel strands, helical parameters. This all I'll be discussing with you over here, that whatever we know about the DNA is basically now with the base we are having because of Watson and Crick. Okay, now let me tell you. Now this over here, basically this DNA comprises of 3 things. What is DNA? When they try to find out this DNA, exactly what it is, you will be finding deoxy ribonucleic acid, right? This is the full form over here for DNA. So this structure of DNA is containing first the pentose sugar molecule, second a phosphate group, third nitrogenous bases. These 3 things are basically constituting together to form a DNA. Now see the structure of this pentose sugar. This is very important to understand over here. What is pentose sugar basically? Pentose means 5. Where is the 5th one? This is the 5th one. So this bond, this bond, this bond. You just have to understand this. Nowhere you have to make this type of things. Just understand it. 1 carbon, 2 carbon, 3 carbon, 4 carbon and 5 carbon. With every carbon you will be finding one OH molecule will be attached. कार्बन की वैलेंसी 4 होती है, अगर नहीं भी पता डजेंट मैटर, ये आपको सीखने का नहीं है, सिर्फ आप डायरेक्टली इसको समझो, राइट? इट डजेंट मैटर इफ यू डोंट नो दैट क्या वैलेंसी. वैसे तो 9th क्लास एनसीईआरटी यू हैव टू रीड, ठीक है? देयर यू विल बी फाइंडिंग, एवरीबडी मस्ट बी नोइंग दैट कार्बन हैज 4 वैलेंसीज. 4 means 4 bond it will be having then, right? One with OH over here, one with H, one with OH over here, one with H, one with OH over here, one with H, one with OH over here, and over here what you will be finding, one bond is also there, only one bond will be happening, so OH over here you will be finding, or sorry, H over here. This you will be finding like H, right? So don't see all of it right now, just see this particular carbon molecule over here, just see this particular carbon molecule that is present at the 2nd number. Now what happens over here, that it is connected with the OH molecule over here, right? OH and one with H. ठीक है, ये आप देख रहे हो. So when the second molecule is connected with the OH, oxygen, and this hydroxyl group over here, OH, that is known as your ribose. Right? Whereas if you will be removing this O from here, that means now C will only be attached with H, not OH. Oxygen has been removed. That means this is now deoxyribose. And this is the major difference over here. I am telling you, when I have removed O from here, it becomes deoxyribose. And when it is OH is present, it is a ribose. Okay, that is how you will be finding. In the end, I will be telling you the difference between DNA and RNA. DNA is a deoxyribo. So deoxyribo pentose sugar is basically this DNA only. And when we will be seeing ribonucleic deoxy, हम हटा देंगे यहाँ से, तो it will become RNA, which I will be telling you later on. ठीक है, तो this pentose sugar plays an important role. I'm really sorry this has been erased, but pentose sugar I was telling you. So for your examination, you have to remember this structure only. Just this structure you remember for the examination. O. And this is 2 groups. So this is over here H, and this is over here carbon, like this. 5 carbon over here. Pentose sugar. Very important. Now pentose sugar you are finding over here, guys. Second, I told you that there is a phosphate group in the structure. What is this phosphate group? That is H3PO4 negative. H3PO4 negative over here. H over here, H over here, H over here, and O over here you will be finding. Really sorry, this is OH, right? तभी तो 4 होंगे, not H, OH. OH, OH, OH. Right, and this is negative because hydrogen is not attached. As it is, you understand this. 4 oxygen, 1 P, 3 hydrogen over here, H3PO negative. This is how you will be finding this phosphate group. So this phosphate group will be attaching where? This will be attaching over here with the 5th carbon over here, guys. This is the 5th carbon molecule you are having. The phosphate group will be attaching over here. And in the structure, we will be just showing by the P. By like this, we will be showing in the examination by P. That's why it is simpler. You don't have to go into the chemistry of it. Then there is nitrogenous bases. Nitrogenous bases over here. What you will be finding in the nitrogenous bases, guys, basically, this nitrogenous bases consist of 2 things over here. Let me tell you. So nitrogenous bases you will be seeing in the form of 2 basically. That is purines. Achha, nitrogenous bases you must be understanding that these are the molecules or these are the organic compounds which are consisting of nitrogen. Mainly nitrogen will be there. Right, so that's why nitrogen is very important for us. Nitrogenous bases. Right. I'll be showing you the compound formation also. You don't need to learn that, but I'll be showing you. They are divided into purines and pyrimidines. Over here you will be finding that purines are basically your A and G. And this you have to learn. These are the base pair, you can say, bases. Nitrogenous bases I have told you. That is adenine, guanine, U, C, T, uracil, C से check करेंगे हम, thymine, and C से is your, let me check over here, cytosine, guys. C से था आपका cytosine, right? Let me show you too so that you can understand in a better way. Cytosine, purines and pyrimidines, right? 3 I have told you over here. Let me show you first and then we'll be discussing further. See, first of all, this pentose sugar, I hope you have understood. O, 1 carbon, 2 carbon, यहाँ से शुरू करोगे, 1, 2, 3, 4, 5 over here. And if OH is attached at the second carbon only, you have to check, right? And you have to follow this second carbon. Ribose अगर नहीं है यहाँ पर second carbon में, तो deoxyribose. Then this is the, देखो, this is the structure. I don't want you to learn the structure. There is no need, neither it is asked in the examination. Adenine, guanine, these are your purines. What is there? Nitrogen. They are containing these organic molecule, nitrogen bases. This is cytosine, thymine, and uracil. Now, one thing you will be finding that in DNA you don't have uracil. You have only thymine and cytosine, and you have adenine and guanine. Whereas in RNA you don't have thymine, or you don't have this thymine res. You have this uracil. See guys, over here you check this now, how this nucleotide is connected. Now I have told you that this DNA is having a structure. In this structure you will be finding pentose sugar, you will be finding phosphate group, and you will be finding nitrogenous bases. But how is this connected now? This is connected in the form of, you will be finding that this is your pentose sugar, suppose. Alright, what you will be finding over here, that this pentose sugar, देखो यहाँ पर इसके फर्स्ट कार्बन के साथ there will be nucleotide which will be attached, right? And fifth carbon के साथ there will be phosphate group which will be attached. Understanding? Now there will be another this particular group will be coming over here, and with this again you will be finding nucleotide will be attached, and with the fifth carbon phosphate group will be attached. Now this phosphate group will be having the connection with the third carbon over here, guys, like this. Understanding? Again there will be one more phosphate group will be there, nucleotide over here, and this phosphate group will be attached like this with the fifth carbon. This is the fifth carbon, this is the connected over here with the phosphate group, third carbon over here, and then they will be having the attachment like this with this particular group. This is forming your base, and these are your nitrogenous bases over here. Understanding? Now see this structure. This is sugar. With the fifth molecule, this phosphate group is attached, and with the first carbon molecule, this nitrogenous base is attached. Now you don't have to show this like this in the examination. The way I have shown you, you can show like this. Otherwise, there will be no need to show also this type of structure, right? Now let me tell you how these things are happening over here. So simple, what you will be finding over here, that when we saw the structure, right now just try to understand this, है ना? Over here like this, pentose sugar, pentose sugar, pentose sugar. ये कहीं दिखाना नहीं है, समझाने के लिए कर रहा हूँ यहाँ पर, ठीक है? Pentose sugar. Over here you will be finding pentose sugar, pentose sugar, pentose sugar like this, and pentose sugar like this. ठीक है, ये आपकी pentose sugar है. देखो यहाँ पर क्या होगा आपका, with the first carbon molecule there will be nitrogenous base attached. ये आपका first carbon molecule nitrogen base will be attached. First nitrogen base, first nitrogen base, first nitrogen base. ये उल्टा है, उल्टी strand है, opposite nitrogen base, nitrogen base, nitrogen base. So what you will be finding that there will be a connection between these two, either double bond or triple bond. This I'll be telling you later on when we'll be exploring in this particular lecture only. This what you have found, phosphate group. Phosphate group will be having connection over here too. Phosphate group, phosphate group. Likewise there will be phosphate group, phosphate group, phosphate group, and a phosphate group. There will be connection like this you will be finding. Okay, now this is nothing but this helical structure. Suppose I have shown you like this. This is nothing but these ladder type of thing. What Watson and Crick have found, a double helical. Now this will be converting like this over here, like this you will be finding. Converted double helical type of structure you will be finding. This you have opened it up now. Now you see, let me show you over here. You see, try to see, guys. This is phosphate group over here. This is one chain of phosphate group over here. These are nucleotides. I'll be telling you what are these nucleotides. Already I have told you AGT. All nucleotides will be only AT or basically AG purines and pyrimidines. TCU. U only in case of RNA. When there will be RNA, there will be no T. When there will be DNA, there will be no U. Right? This is one more difference. I'll be telling you the difference between RNA and DNA. This is pyrimidines. Right? This I have told you. I'll be telling you later on how these two bondings are happening over here. See this again. Suppose this strand you want to open up. This is the phosphate group attached over here. This is the pentose sugar. This is your nitrogenous bases connected with the other part over here. Guanine over here, cytosine over here, right? Now see this one rule over here, that is Chargaff's rule, guys. Chargaff's rule. Chargaff found that nucleotide bases are not present in equal amounts over here. The base composition varies between species. No need to remember, but you try to understand this statement that nucleotide bases are not present in equal amounts, right? Key finding is purines total A plus G, C and T total pyrimidines के equal होगा, right? So adenine ratio thymine is equal to 1 and GC ratio will be equal to 1. Over here you will be finding that A, G, T, C in the case of DNA, A, G, U, C in the case of RNA. This is the case of DNA. What happens? Purines and pyrimidines get together over here, like AT, like GC, like AU and GC over here. AT is having double bond, whereas GC is having triple bond. These bases will be connecting with itself always. Right? Write this, you will be finding AT, GC, AU, GC. So see the double helical structure given by Watson and Crick now. Double stranded structure I have explained you very clearly over here. 2 polynucleotide. Acha, this particular one, this particular P over here, N over here, this is called one nucleotide. This is a nucleotide. When several nucleotide will be joining together, it will be forming a polynucleotide chain. Polynucleotide chain. So that's why it is written as DNA consists of 2 polynucleotide chain, means because one from here polynucleotide chain, from one over here polynucleotide chain, and then revolved like helical shape may they will be over here. So that's why it is written wounded each other in a right-handed double helical structure. Anti-parallel orientation means if the strand is 5:3 or like this, it will be going 5 dash means that the carbon molecule will be attached, this phosphate group will be attached as 5 dash, like this 5 dash, whereas in the opposite strand you will be finding that it will be attached to the third strand over here, 3rd to 5 it will be moved like this. Okay, if this is moving like this, then it will be moving like this over here. Okay, opposite strand will be there. I'll be showing you, don't worry, I'll be showing you in the animation also. Backbone structure. Backbone structure is formed by the phosphodiester bond between sugar and phosphate group, which you have seen like this. This is making the backbone, and this is the nucleotide in connection with each other. Nitrogenous bases are oriented toward the center. I hope you are understanding right now. Complementary base pairing will be there, like A to T and G to C will be happening. 2 hydrogen bonds, 3 hydrogen bonds. Always one purine paired with one pyrimidine. Right, like this it will be maintaining. Dimensions of DNA helix like this you can remember, but no need. Ahead I'll be telling you obviously. Okay guys, now you understand that 2 animation I'll be showing you of the nucleus structure which we have read and DNA structure which we have read right now, right, which we have understood. After that I'll be telling you about the DNA replication and gene to protein, how it gets transformed. Okay, see this guys, one good animation video you will be finding over here. I am utilizing YouTube for it. You don't have to search it. See over here, one plant tissue we will be seeing, and any cell we will be opening now. Plant cell, this is one plant cell. We are going ahead in this plant cell, right? In this you will be finding this particular cell. You can find mitochondria, you can spot Golgi body, endoplasmic reticulum, and then we will be going to the nucleus. Nucleus is having nuclear pores. So just observe this particular animation over here. These are the nuclear pores which I have shown you. These are protein pores you will be finding. Length also you try to find that 5 to 25 micrometers. You will be finding somewhere 5 micrometer and somewhere 25 micrometers. You will be finding this diameter over here, right? This is nuclear envelope which we have understood. Nuclear envelope means outer membrane and inner membrane, double membrane you will be understanding. Everything, the liquid inside the nucleus will be nucleoplasm, right? And this thread-like structure you will be finding chromatin, and this inside dense material you will be finding which is membrane-less, that is nucleolus you have seen, right? Nuclear envelope you will be finding over here. See this now, nuclear envelope. This is bilayer, phospholipid layer, outer layer and inner layer. Then nucleoplasm, cytoplasm you are finding over here. Nuclear envelope, again they are showing you this. It is also known as karyotheca. Okay, over here see this particular trilaminar protein, lipids and protein over here. Trilaminar you must be knowing right now, right? The phospholipid layer you will be finding over here. See it clearly guys. Okay, now see like the spread over here. Just wait. ठीक है, over here you will be finding outer membrane you have seen. Now these are nuclear pores. From here, whatever RNA, I'll be telling you about this, how proteins are getting formed. This we have to study right now. Right? Just observe this thing. This is the way this RNA will be coming. Proteins will be going, required proteins in the nucleoplasm, it will be going. Okay, this is your protein structure they are trying to tell you. Over there, now you will be finding nucleoplasm, and nucleoplasm will be containing, I have told you, a lot of enzymes which are needed for the functioning of this particular nucleus. Okay, and this is a nucleolus type of structure you will be finding. In this you will be finding देखो ये nucleoplasm में you will be getting all these enzymes, DNA polymerase. ठीक है, all this I'll be telling you what is RNA polymerase. This is I am going to tell you in that DNA replication which we are going to study just now. ठीक है, यहाँ पर just you try to focus upon over here. This is a nucleoli or nucleolus. Over here, what will be happening? I have told you rRNA will be built up over here, and the ribosome formation you will be seeing over here. ठीक है, so this is over here you will be finding with respect to nucleolus. Next animation you see on the structure and function of DNA. I am utilizing one of the video by Score Booster Academy over here. Right, they have made this animation. Just see it. Structure of deoxyribonucleic acid over here you will be finding. So this way, this is the helical structure it is. Right, now try to observe this particular band which you are seeing. It is consisting of what? You know that this is consisting of my phosphate group and pentose sugar. Right, this is phosphate and pentose sugar, and these strands which you are seeing, these are nothing but your nuclear base, nitrogenous base. So there are three components in DNA which you always have to remember, because this is what you will be writing. So nucleon, just wait over here. James Watson and Francis Crick model you will be finding over here, a double helical structure like this, and we will be focusing upon this Watson and Crick particular structure. Right, this is a helical nature you will be finding of DNA. 4 nucleotide bases. What are those bases? Purines and pyrimidines. Purines, what are those in purines? A, G, and pyrimidines you will be finding in a DNA, T, C, and in the RNA you will be finding, except T will not be there, uracil will be there, and C will be there. Right, so adenine, cytosine, guanine, and thymine. But in RNA you will be finding uracil also. Okay, see this sugar, phosphate, backbone, and nucleotides like this. And I have told you that the arrangement, these will be attaching with one thing only. Okay, just see over here. So this is the way it will be coiling up over here. Hydrogen bond will be there. That means adenine and thymine, I have told you, always adenine will combine with thymine. This is the fixed thing over here which we have found. These things are existing as it is in nature. We have understood it, that what are these. So adenine and thymine are having double bond structure, double hydrogen bond you will be finding. Okay, like this they will be joining over here. I hope you are understanding this. Beautifully shown. One thing you will be finding, that somewhere when these particular DNA segments are there like this, you will be finding one is a major groove and one is a minor groove. Not very important, but major groove and minor groove also you will be seeing in the PPT also. I have added this major groove and minor groove. Okay, over here you will be finding this. Supercoiling is happening. Eukaryotic cells you will be finding, and this I'll be telling you in chromosome. Dekho, the structure which I was showing you. When you will be opening it up, the dense chromosome, the dense DNA has been condensed in by the histone proteins. They are showing histone proteins over here. So it is doing the job of condensing this DNA into a small package of chromosome. Right, over here I'll be telling you this later on also, there what is histone and all of these. Right, cell dividing we will be understanding. So I hope you have understood this. Okay, chromatin and how it will be dividing, this is now not the scope. This we will be seeing. I'll be telling you later on in detail about the cell cycle. Okay, so 2 things I have shown you from the animation also. Animation is just for your understanding. Ultimately you have to write in the examination in 2 pages for 10 marker and in 3 pages for 15 to 20 marker over here. Right, so keep that in mind. Now we will be moving ahead and we will be understanding that what is the mechanism of DNA replication. how DNA is getting replicated, guys. This now you try to understand. This is the next topic you have under this. Structure of this happened, nucleus happened. You have understood the theories evolving the DNA structure. Then DNA structure you understand under Watson and Crick. Okay, now DNA replication, how it happens basically, how DNA is converting their copy into an identical one with certain modification. This you have to understand now. Okay. Now the question over here was regarding that we have understood the DNA structure. You will be finding that they have understood this DNA, that this DNA molecule over here is responsible for giving basically the existence to us. The appearance is given by these molecules over here, and these molecules are found everywhere in the organisms, living beings, and these can replicate themselves. That means the offsprings are having the same copies of these DNA. Also in an organism, suppose in you will be finding that all trillions of cells will be having the same copy of this DNA. That means that this DNA is replicating itself. So the question in 1953 emerged that how DNA basically replicates itself over here. The question was this in 1953, that how basically this DNA is replicating. Now the structure, a lot of research have happened. Okay, we have understood the structure. Now let us explore that how this DNA is getting replicated, because this is very important to understand. By this understanding only, today what we can do in a genetic engineering is with the help of the 100 years old this experiments which have happened. All right. Right now we can understand that what are the genetic diseases and how we can cure it. Right now we can do certain BT cotton, GM mustard, like this we can improve our crop variety too. And this is the need of the future also, guys. We are understanding that now we can create these superhuman babies, right? Where we can select gene, where we can like select the genes which we want over here. You will be understanding CRISPR is based upon this DNA understanding only. So this is very important for us to understand the inheritance laws, for the heritability over here when we will be going ahead in this unit. So this is very important. So when they were understanding, when these scientists were understanding about DNA replication, the three theories were prominent over here. One theory was a conservative theory. Conservative, right? This is also known as conservative replication. Then there is second, dispersive replication. This is exactly how you have to remember, dispersive replication. And third is a semi-conservative replication. Semi-conservative replication. So as per this conservative replication method, what they suggested, that suppose this is a DNA strand over here, right? This is a DNA strand. As per this conservative model, they were saying that this DNA is completely replicating itself like this in the same manner and same procedure. You will be finding that this has been replicated like this. This has been converted from this to this over here. The dispersive method, replication method, was suggesting that what is happening, that over here, this DNA molecule, like this which you are seeing, is getting dispersed into some small pieces. So what is going to happen? So over here you will be finding that they will be cutting into small pieces. So one cut over here, like this, they will be getting fragmented over here, and each fragment will now be making its copy. So over here copy will be made, fragment copy will be made over here, right? And now what they suggested, that now these copies will be joined together like this. And how it will be joining? It will be having fragments of this particular part also and this part also. That means the replicated one and the new one and the old one. This we can call the old, and this the new one over here. So this is the combined DNA which they have formed over here. Now both of these things were rejected, guys. After one experiment done by two scientists, name I'll be telling you, Stoll and Mellison, you will be finding. I'll be telling you also in the PPT exact name with correct spelling. I'll be telling you. Okay. So they gave this semi-conservative replication. They proved it by the experiment, which basically thwarted these theories which were going on. And this is the accepted model of DNA replication, semi-conservative. What is semi-conservative? Basically, semi-conservative is nothing over here that they were saying that what is going to happen. Let me explain you. See over here, like for example, this is your DNA. This is your DNA over here. Semi-conservative, I am telling you. A, T, G, C, A, T, G, C, A, T, G, C, A, T. Suppose this is the model. What is going to happen in semi-conservative, that this 5' to 3' line and 3' to 5' line, always opposite, I have told you already, right? They will be opening up. That means this strand will be opening up like this. This is 5' to 3' line, this one, ये जो line है, ठीक है, and the separate line will be opening up like this, 5' to 3' line. All right. Now what is going to happen over here? Suppose this was AT, GC, AT, GC, AT, GC. Okay, A over here, this is nucleotide A. Now this has been opened up like this. This has been again opened up like this. So what you are understanding, that these two strands have opened up. Okay, this model which you are seeing over here, this have opened up like this, and the enzymes will be working. I'll be telling you. Simply I'm telling you right now how the semi-conservative, what does it mean? Now this part over here will be replicating like this. This part will be containing the new strand like this. Suppose this was A, then AT, G, C, A, T, G, C, A, T. That means this is your new strand which has been created. Similarly over here with this strand will be coming into picture, and they will be same as you will be finding. This is 5' to 3', and this is a one new line will be created over here. That means two DNA is formed out of one DNA, where it is conserving one part as it is from the parent DNA. That's why semi-conservative means. Why? Because half of it is new added, whereas in conservative the whole DNA is getting replicated. This is not the case, which was found out by I'll be telling you the name of Stoll and Messel, something you will be finding. See over here guys, everything I have given you in the notes. More than this notes, you will not be needing anything. I have updated it, and you can utilize it whenever, like anytime you can utilize these particular notes. Okay, I have updated it to the most recent. Okay, and these things will not change, है ना? So over here, modes of DNA replication, I have told you: conservative, dispersive, and semi-conservative. See, what I have told you is written over here. You can pause the video and remember, and you have to write like this, है ना? This was for explanation. So the scientist of semi-conservative model were Michelson and Stoll, right? Michelson and Stoll, you will be remembering, in 1958 gave that the actual mode of DNA replication in living cell is a semi-conservative model. And this experiment completely revolutionized this genetic research, guys. Why? Because then they understood that DNA can replicate itself by separating its portion also. That means DNA can break over here. And what experiment they did, now it has not been asked till now, but be on the safer side, and you have to prepare in the most right manner. So I would suggest you that you just understand it with me, and this is the way you have to write it in the examination. 2-pager information, though they will be asking you this about this experiments in 2 parts. So 1-pager information you need for the examination for this experiment also of Michelson and Stoll experiment. This is also known as the most beautiful experiment in genetics or in biology. Over here you will be finding what they did, that they again took this E. coli bacteria. So over here you will be finding this E. coli was grown for many generations in a medium containing 15 nitrogen. 15 nitrogen means the atomic number is 15 over here of nitrogen. It is a heavy isotope of nitrogen. And nitrogen is particularly 14, but 15 why? Because this is a heavy isotope of nitrogen. So DNA became denser because density of 15 DNA was 1.724. No need to remember. Density of 14 nitrogen for the other isotope of nitrogen is 1.7. So what you have to understand over here, when we were growing these E. coli, you will be finding that there were 2 types of E. coli which were grown. One that was in heavier density because of containing 15 nitrogen, and one of lighter density because of containing 14 nitrogen. Now separation by density gradient centrifugation means now we will be separating it. This is a mixture over here, and if we want to separate the lightness and heaviness of any particle, we should use centrifugation technique, right? So similarly, centrifugation technique was used over here to distinguish between heavy particles and over here light particles. So distinct heavy DNA band over here we have got. Suppose this is the test tube and we have do the centrifugation. So over here are heavy DNA accumulated and over here are light DNA accumulated. Now labeled E. coli cells transferred to 14N medium and allowed to replicate. That means over here the light over here, the E. coli was transferred to the 14 nitrogen, lighter nitrogen medium over here and allowed to replicate over here, right? See what they are trying to say over here. You try to understand over here again. देखो कुछ नहीं कर रहे हम. How first we were taking the E. coli over here. E. coli लिया. Now E. coli, what we did over here, that we treated them with 15 nitrogen over here, right? Because of that, what will be happening, that in their DNA heavy isotope will be coming, and in some E. coli there will be 14 nitrogen. So this is a mixture which have been created. I have to separate the mixture. I'll be doing this this particular centrifugation technique over here. What they will be doing, it will be separating the particle over here, separating the heavier one and the lighter one over here, like this. Now what they have done, that they will now allow this particular 14 and lighter particle to replicate over here. Now you try to understand that, see over here, this particular labeled E. coli means that this particular 15N DNA will now be transferred to the 14N medium. Understanding? So this is your already 14N medium, nitrogen is present, but now I am transferring my E. coli with 15N. What is going to happen? Now when they will be reacting with this particular 14N, that means either 15N should come, 14N should come, or what in mix between 15 and 14N should come, right? This is what is going to happen with respect to when E. coli will be replicating. Okay, 14N should also be seen, 15N should also be seen. This is the case, that what will be the result of this particular thing is going to be. So as per conservative, if you will be finding, as per conservative model, E. coli is having 15 nitrogen, but you have providing 14N solution over here, 14N medium over here. As per conservative model, what should happen, that this 15N should give only 15N heavy isotope DNA E. coli. It should replicate and should have only 15N. But this was not the case which was seen in the result of the progenies of E. coli. When E. coli replicated, what you will be finding, you will be shocked to find that over there you will be finding that there is a mixture of 15N and 14N lighter molecule and heavy molecule DNA which we have found over here. That means this conservative model has been rejected, and the two models, the doubt is on the two models, semi-conservative and the dispersive model, because in both the models you will be finding that half of them were opening up and dispersive fragments are opening up. So after one generation, DNA had intermediate density. Why intermediate density? Because it will be having hybrid of 15N and 14N strands. So it supports semi-conservative replication and also dispersive replication. But when we replicated this again, what you will be finding, when this E. coli replicated again, you will be finding that half of the DNA was hybrid, that is 15N and 14N, and half was light only. That means it is supporting your, over here, it is supporting your semi-conservative model, like this, because half will remain 14N only and half will be containing 14N and 15N second generation. This is exactly how you have to show in the examination, guys. Till now has not been asked, but can be asked anytime. You are in the preparation phase, you will not leave any part. And when I have told you that out of 8 units, out of 7 units in paper number 2, 5 units should be on your tips. So you will not be leaving any stone unturned. In rest of the 2 units, you can only do what is being asked. But over here, what is not being asked also, I will be letting you do. Okay, this is my job. So right now, after one generation like this, you will be writing. After 2 generation, what will be happening? A hybrid and half was light only, 14N only. After 3rd generation, you will be finding one will be remaining 3/4 will be 14 and 1/4 will be hybrid. That means the ratio is going down, and two strands semi-conservative, the earlier strand of 14N is also being utilized, and a hybrid also coming over here. Understanding a new hybrid is also being created over here. So this observation is supporting what your semi-conservative model over here. So the experiments by Michelson and Stahl provided definite proof that DNA replication occurs in a semi-conservative manner. Each daughter nuclei contain one parental and one newly synthesized strand over here, right? So this is what Michelson and Stahl experiment. You will be writing it in simple understanding. Based upon your understanding, this you have to write over here. Okay, so this, a lot of things you have understood in DNA part. This is DNA replication we are doing. Now the methods of semi-conservative DNA replication also you have to understand that how this DNA replication is happening over here. So to understand this, there are certain procedures you will be finding, and there are total 7 procedures. I am going to tell you that how this is going to happen. Very easy to understand, and I'll be showing you the animation of this also. Okay, therefore try to understand that there are certain enzymes which are going to be utilized in DNA replication, and where these enzymes will be present. These enzymes will automatically be present in the nucleoplasm. Now is the right time to understand these enzymes. Enzymes like helicase you are going to find over here, guys. Helicase. What helicase will be doing? It will be opening up. It is like a molecular scissor. Okay, helicase. It will opening up the DNA. Helicase. Then you will be needing topomerase or SSP. Single strand binding proteins over here. That I'll be also telling you where they will be working upon. Then you will be doing RNA primer and DNA primer over here you will be utilizing for pasting it, or DNA polymerase you write over here, DNA polymerase. One more will be there, that is a ligase, and I'll be telling you now how this is going to happen. See, there are certain stages over here. Try to understand, then I'll be showing you the एनिमेशन ऑल्सो. पहले थोड़ा सा दिमाग पे थोड़ा जोर लगाओ, फिर एनिमेशन से क्लियर चीजें होंगी. अदरवाइज एनिमेशन आपको पता ही नहीं लगेगा कैसे यूटिलाइज करना है. ठीक है, एंड नो नीड टू सर्च मोर गाइस. इफ यू विल बी सर्चिंग मोर, यू विल बी वेस्टिंग योर टाइम ओवर हियर. ठीक है, यू कैन सर्च इट बट नॉट डिसप्रोपोर्शनेटली. ठीक है, दिस इज योर डीएनए. ओवर हियर आई एम कॉलिंग दिस 5' एंड टू 3' and this is opposite. This is the rule 3' to 5'. Now suppose over here this replication has to happen of this particular strand. See, understand carefully. So if replication has to happen, suppose I am taking this fragment only, the separation or this particular fragment only I am showing you with these 3 nitrogen bases how these are going to replicate. ठीक है, यहाँ पर 1-2 bases और लगा देते हैं हम यहाँ पर. ठीक है, so that हमको थोड़ा सा और ज्यादा देखने को मिले. तो यहाँ पर how it will be happening over here, helicase will be working, and this I am showing you guys. Helicase is going to work over here, and this strand helicase will be opening up this strand over here like this. So this strand over here has been opened. So this is the line from 3' to 5'. Understand, this is 3' to 5' over here, this one, and this is 5' to 3'. And this helicase have cut it open in 2 parts, right? And with the help of these SSB proteins, topomerase, they will not come back again. So they are at stiff position over here. And what are their strands like? Their strands have broken down into like this, suppose. Okay, how many I have drawn? 5. So these are their strands. Okay, suppose this was A, T. You know, always G, C. A, T, G, C, A, T. These are your nitrogen bases. What is going to happen now is, now first of all there will be RNA primer will be at picture. RNA primer will be coming and will be marking this particular site that from here this DNA strand has to get replicated. And DNA RNA primase will be utilized over here. Primer and DNA polymerase will be utilizing over here. And this DNA polymerase will be bringing one new strand over here with the blue I am showing. They will be bringing one new strand over here with 5' and to 3' end, because this should always be opposite. And what they will be bringing over here, they will be bringing the new fragment over here of this particular nitrogen bases. So A will always be connected with T, G will always be connected with C, A with T, G with C, A with T, like this. And over here in the end they will be utilizing this ligase protein, so that they can merge with each other like this. And this is called a leading strand. This particular strand which has been created, this is called as a leading strand. This is the terminology which you have to utilize. Now there is one more formation which is going to happen over here, that is with respect to, अभी ये मैं हटा देता हूँ, ये समझ में आ गया होगा आपको, ठीक है। ये यहाँ पर एक स्ट्रैंड बन चुका है, ठीक है। This strand has been created over here, ठीक है। This is not complete yet. Now over here you will be finding that this 5' to 3' end, that means over here opposite should come 5' to 3' end. But what happens over here, that this becomes our lagging strand and it does not go as smooth as this, like we have seen from 5' to 3'. When this is a lagging strand always being created by in fragments. So it will be marking, this RNA primer will be marking over here and it will be creating one fragment like this. Then it will be coming over here marking and it will be creating fragment like this. So 5' to 3', 5' to 3'. So these small fragments like this, they will be joined in the lagging strand, and that's why it is known as a lagging strand. Right now just understand what I am saying. When I will be showing the PPT also, images also, and the animation also, you will be understanding. These fragments were founded by Okazaki you will be finding, and that's why these fragments are known as Okazaki fragments also. Okay, and this is the way this particular lagging strand is being created. So semi-conservative, too new, too old over here. I hope you are understanding this. Let me show you now. See, understand this. And this process is known as initiation of replication first. What is going to happen, that they will be getting replicated over here. They will be getting ready to get replicated. Unwinding will be done by DNA gyrase and DNA helicase. Unwind the double-stranded DNA. Then what will be happening? Stabilizing of single strand. What is the single strand which has been formed over here? They will be stabilized. How they will be stabilized? By utilizing these SSPs. Single strand binding. This should be highlighted in your answers, guys. Okay? Then primer synthesis will be happening. That means primase and RNA polymerase synthesizes short RNA primers complementary to the template strand over here. And strand synthesis by DNA polymerase then. DNA polymerase will work over here and will be 5' to 3' direction. They will be forming this particular direction in no breaks. Leading strand synthesized continuously like this. Lagging strand synthesized in breaks over here. 5' to 3', 5' to 3'. So synthesize discontinuously away from the replication fork. Produce short segments like Okazaki fragments, which I have told you. Then what will be happening? The integration, joining of fragments is going to happen. DNA ligase joins Okazaki fragments to form continuous strands. Right? So I hope you are understanding this. See this from this diagram, guys. Like this also you can show. This is C over here first. Let me focus where I want you to see first. So over here, what they will be doing by using the helicase, they will be opening up this like this over here. One end will be going like this. Okay, one will become the lagging strand and one will become the leading strand. What is going to happen? These primaries, helicase, SSPs will be stabilizing it. DNA polymerase will be coming over here like this, DNA polymerase, and bringing a new segment over here. Okay. So if this is a leading strand, the continuous chain will be formed. If it is a lagging strand, there will be breaks over here like this. Okazaki fragment. Okay. Now see the animation and it will be clear to you. See over here, guys. Now DNA replication. Okay. ध्यान से देखना. Just wait. So this strand is opening up with the help of topoisomerase and with the help of your helicase. I hope you have remembered that. See ATGC over here, 5' to 3' line. This is a parent DNA molecule, 5' to 3' opposite. Now they will be opening up over here. Who is opening up? Helicase. Because they have written helicase over here. And this is also known as a replication fork. Remember the terminology in the examination. You will not be drawing this all these images. Hai na? See it again. Primase is coming, RNA primase. And they will be forming over here one primer. Why this primer is there? Primer is for identification that from here replication will be done. So see this over here. Now DNA polymerase, RNA primase work is done. DNA polymerase is coming and DNA polymerase is attracting these bases, new bases. And a leading strand will be created without any fragments, without any discontinuity. And this will keep on creating. Whereas in the lagging strand, what you will be going to find that 5' to 3' Okazaki fragments will be formed, and by this way it will be created, because it is 5' to 3' end in that particular thing, right? Same process is going to happen. Okay. Now what will be happening over here? Exonucleus means this will be removing the primase over here, and again DNA polymerase will be coming and adding new nucleotides over here, or adding new nitrogenous bases. Then last DNA ligase will be coming over here, and will be basically doing this replicated strand over here and the semi-conservative method. So I hope you have understood this DNA replication. Now again I am telling you, simple one 10 marker or 15 marker question is going to come. This is enough which you will be writing over here. Okay, this I have already again explained you, and in this points only you will be writing as this. Okay, just copy it as it is with your own understanding, guys. Now let us understand our next important topic, that is your gene to protein expression. Gene to protein expression, right? How this gene is converting to protein over here. You must have understood that this gene is basically the functional unit of DNA, and DNA we have expanded and understood clearly with the theories which involved. DNA is basically coiled up in chromosomes, which you have understood over here. Chromosomes are interned like fiber-like structure in chromatin, you have understood. Chromatin are present inside the nucleus, you have understood this, and nucleus is present inside the cell. This thing you must have understood right now. Chromosome, chromatin we have to study again in detail in that separate lecture, because we have one subtopic as, or subunit as, chromosomal aberrations, that we have to see it in detail, तो वो हम separately देखेंगे. The question is now, this particular DNA which we have seen, how it will be expressing itself, this we have to understand now. Okay, this is, suppose this part of the DNA is functional. If it is functional, means it is responsible for some expression in a particular organism, okay, in a particular species. Now this will be called as gene. Now from gene to protein will be converted. Gene will be making certain protein, for example hair color, eye color, height, something they will be making over here, and this undergoes under two processes you will be finding. One is transcription and one is translation. These two processes happens in the gene to protein process, right? And a very innovative type of question was asked in UPSC civil services in 2025 recently only, you will be finding. They were saying that how nitrogen is assimilated or how nitrogen is absorbed from the atmosphere. They are asking nitrogen fixation from paper 1, and then they are asking that how nitrogen is converted into amino acid. So conversion of nitrogen into amino acid is nothing but the gene to protein expression you will be finding, because protein is nothing but a compilation of amino acids. Protein is a polyamino tide change over here you will be finding, right? Whereas DNA is the polynucleotide chain, right? So these two processes are coming into picture now, guys. और इसी को हम बोलते हैं यहाँ पर पॉलीपेप्टाइड चेन भी. पॉलीपेप्टाइड, एमिनोटाइड नहीं, पॉलीपेप्टाइड चेन. पेप्टाइड्स आर नथिंग बट द कॉम्बिनेशन ऑफ these amino acids. So for this transcription and translation, now you understand over here. I'll be showing you the animation also. I have told you that this particular part is functional. This is for certain expression over here. So what this will be called as a gene over here. This gene is for eye color, suppose. Okay, now what will be happening over here, that this particular thing will be opening up like this, guys, right? And there will be strands. Again it will be opened up over here by the helicase. You will be finding RNA primer will be used over here, like this it will be opened up, and over here one strand will be created. Only one strand will be created over here, guys, like this, and this strand will be known as mRNA. This is your single stranded DNA, which is RNA, ribonucleic acid over here, and M is for messenger RNA. That means this particular coding strand is containing the code of this particular DNA, and it will be now detaching itself and will be moving freely like this. So RNA is single stranded. Also, I have told you if the sequence is AT, A will be combining with G, T will be combining with AT, sorry, A will be combining with T over here, right? But in RNA, T is not there. What is in replacement of T? You have uracil over here. That's why if we compare DNA and RNA, DNA is more stable because of the nitrogenous compound it has, and RNA is not that much stable as we compared it to the DNA. देखो, RNA basically are of different kinds, a lot of kinds as with respect to their function, but the main functions of RNA with respect to you are having mRNA, which is going to create in the procedure of transcription, which I'll be showing you. Then there is one tRNA, which is responsible in translation procedure, which I'll be telling you. And then there is one more RNA, which is important in the protein synthesis ultimately, that is ribosomal RNA. So these are the three types of main RNA. Then there are other types of RNA also you will be finding. So RNA is a single stranded nuclei made of ribonucleotides. It plays a key role in protein synthesis and gene expression. Very important. RNA is structurally similar to DNA but differs in three key aspects. What are those key aspects? One is sugar. Sugar is a ribose over here, not deoxyribose. I have already told you. Strand is double stranded, single stranded in RNA. Nitrogenous bases ATGC in DNA, AUGC in RNA. This I have already told you, right? Easy to understand now. Functions is act as a messenger between DNA and ribosome. What is written in the DNA, who will be taking it to the ribosome, this mRNA strand. Involved in protein synthesis, part of ribosome also over here, carries amino acid to ribosome during protein synthesis, regulates gene expression, right? So three type of RNA you are going to find over here: messenger RNA, transfer RNA, ribosomal RNA. Okay. Carries genetic information from DNA in the nucleus, brings specific amino acids to ribosome. Again, simple. And I'll be showing you more clarity you will be getting over here. Just read it out till then, guys. Okay. So see this procedure over here, where I am letting you focus. First of all, focus over here. Eukaryotic cell. This is the DNA part. DNA part, suppose this is your gene expression. This part of DNA is containing some specific code. How this code will be done? By transcripting it into a strand known as mRNA. So inside the nucleus, this mRNA will be created. It will be going out to the cytosol or cytoplasm, and it will be going to this ribosome. This is the work of ribosome over here. And when it will be going to the ribosome, then over here, these amino acids will be created. Right. So suppose this is your over here. Nucleus is there. In nucleus, mRNA strand will be created, and it will be going out to the ribosome structure, which I have shown you. And that structure I'll be telling you how it works basically. So this is opening up of this. Okay, and what will be utilized? Single strand template RNA polymerase will be utilized over here, and on which coding will be happening, we will be calling it as coding strand. Okay, then this template strand will be on which coding is happening, and this is the coding strand. Coding strand will be going over here. ठीक है, and then this is a template strand which is being created. देखो, इसको समझने के मतलब इसको आपको as it is replicate नहीं करना, but you try to understand over here. This part I have opened it up. Let me use some other pen. This part I have opened it up. Now from here you will be forming this mRNA strand is being created. This is highlighted over here that how it is being created. देखो, A के साथ हमेशा T लगता है, but this is the RNA strand, so U will be coming over here. So AUG, AUC, so triplets like this particular thing will be created. It will be getting this mRNA will be going to the cytosol like this now. It will be going like this, and 3 units will be created over here. 3 units of the coding strand. And these are also known as codons. Right? This tRNA is picking up these amino acids like this tRNA, and these are containing anticodons. This anticodons will be coming over here. Right, and now it will be attached with this, and this way they will be attaching these proteins, and protein layer will be formed. Let me show you in animation, you will be understanding in a better way. See this part now over here, guys, how from DNA to protein is being converted. Okay, you are try to understand. You will go to the nucleus over here. Nucleus, you will be going to the chromatin fiber. Chromatin fiber, you will be seeing chromosome. Chromosome open up. These are histone protein. We will be understanding it detail in the next lecture. This is your one specific functional unit of DNA is GNA, or is gene. gene you will be finding RNA polymerase will be coming, helicase will be coming and opening up. Then over here one single strand, this RNA will be formed up over here. The code will be taken up by mRNA. Okay, this code will be taken up now. Where? To this particular part, to the cytoplasm. So this will be moving out from the nuclear pores. अब nuclear pores का importance समझ में आ रहा है. Now this will be going to the cytoplasm. This is the ribosome unit. अगर eukaryotes है तो 60s I have told you. Above one 60s, sorry 80s, 60-40. This is tRNA over here you are seeing. These are anticodons present over here. These are codon, anticodon, fixing will be happening, and these way proteins are getting joined. These are amino acids are getting joined. And that is how a polypeptide chain is getting formed. This protein will be going and will be basically translating itself over here, right? So this is what the procedure for translation and transcription you have seen. Translation is basically just creation of this mRNA. This is translation. And this is transcription. Translation is that this formation of this particular polypeptide chain. So like this you will be finding now over here protein synthesis, how it happens over here. Okay, over here you will be finding first transcription from DNA to mRNA. Like this you will be writing as it is, guys. Same things are written again and again. Then you will be finding over here that how this is going to happen in the next process. Translation from mRNA to protein will be happening, and these particular things will be utilized over here. All we have discussed already, right? Translation, what happened? Activation of amino acid transfer to tRNA, chain initiation, chain elongation, and chain termination you will be finding. Okay, so when this chain initiation will be having, this is the larger unit of ribosome. So over here A, P and exit part will be there. tRNA will be coming over here. This will be the site of attachment, and this will be the site of exit over here, termination over here. So that's why initiation, elongation and termination, now you read, you will be understanding it, guys. Okay. So this is you will be finding with respect to gene to protein also, right? Now in this particular lecture we have covered a lot of things over here, guys. Okay, this has been a long lecture. We started off from understanding this nucleus, and what we're understanding about this, that we will be understanding the structure and function. Okay, keep it fixed. 3-4 things I have told you over here, right? After nucleus we have understood that how DNA structure theories emerged over here. DNA theories we have understood, first, second theory, third theory, and over here fourth theory with respect to Watson and Crick. Over here Hershey and Chase, I hope you remember. Avery, McDonald and McCarthy experiment we have understood, and Griffith experiment we have understood. I hope you have not forgotten it. Take a McCarthy, over here Hershey and Chase, Watson and Crick. And over here also then I have told you that in 1958 for DNA replication, after this theory you have understood the DNA structure completely, and what does DNA structure composed of, we have seen in animation also. I have told you what to write also, right? Elaboratively. More than this, if you are investing your time, you are killing other subjects time also. Okay, so don't just have faith and try to revise it multiple times. After DNA structure you have understood DNA replication, and in this the major job was done by your the name always I forget, Messel something you will be finding, right? And they have done this experiment and proved that semi-conservative method is right over here, right? I hope you remember the name. I always forget it no matter how many times I read it. Okay, there is something with that name. Okay, after DNA replication we have seen that how this DNA is replicating, and then we have found that RNA, or how this protein to gene to protein synthesis is happening. Gene to protein synthesis is happening. The role of RNA we have seen over here, right? So this way we have understood this particular topic. So this topic of synthesis, structure, function of genetic material, we have done it elaboratively at the right time over here, and this cell structure function also we have done. Now in the next class we will be moving forward with this particular chromosome understanding and cell cycle also I'll be telling you. Then a separate lecture for chromosome aberration, and then this is linked with polyploidy, euploidy and aneuploidy. This is the importance of understanding a subject. अगर आप ऐसे करोगे तो ऐसे नहीं हो पाएगा, वो रैंडम चीजें, क्रोमोसोम को कब समझना है ये आपको पता होना चाहिए. ठीक है, एंड देन क्रोमोसोम व्हेन यू आर अंडरस्टैंडिंग इट इज लिंक्ड विद दिस, नॉट दिस पर्टिकुलर फिफ्थ, ये फिफ्थ हम बाद में भी कर लेंगे, ठीक है. Fifth we will be doing when we will be picking up this mutation, and then these are two secluded units which we have, subunit which we have to do. Most important unit, and likewise if you are feeling that you are getting like confident in it, do let me know also, do show me your notes. In the enrichment lecture we will be further elaborating these things and rectifying these things only. Okay guys, thank you so much for being patient, and I'll be meeting you in the next class now.